Our next speaker will be Viraat Goel. Viraat is a very brilliant graduate student mentored by Hansen of MIT Bioengineering. He's going to talk about a very interesting and very exciting technology that we can use super high resolution microsite technology to map the gene regulation dynamics. So, stage is yours.
Thank you, Bo, for that introduction. Thank you all for being here, and many thanks to Melina and Casper for the invite. I'm beyond honored to be able to represent the Hansen lab here at the V2F Symposium. And so I don't want to keep you from lunch too long, and so we'll dive right in. And so as you all are very well aware, a very small fraction of the human genome is actually protein coding, only about 2%, which is perhaps then not a surprising fact that leads to the vast majority of disease-associated variants in humans arising in non-coding parts of the genome. This is true for a wide variety of different diseases. I'll be talking later in this talk about metabolic diseases, and so empirical evidence for the fact that the vast majority of variants that impact metabolic diseases do lie in non-coding regions has previously been found, and introns happen to be especially prevalent when it comes to these disease-associated variants. And so when we think about a V2F or variant to function framework, and we want to connect these variants to the function that they have, it's important for us to connect them to the regulatory elements that they impact. Enhancers are one of the primary units of mammalian gene regulation, and they can be proximal to or distal from the genes that they impact. And so beginning to stitch together how variants impact enhancers and their ability to activate gene expression is an important step in using the V2F framework to connect variants to function. Unfortunately, however, 1D genomics methods are ill-equipped to identify enhancer gene interactions. And so here I'm showing you a prototypical example of the c-Myc locus, where we have two different enhancers, 1.7 and 2.1 megabases away. And so in a wild-type condition, it's known that enhancer 1 loops to interact with the c-Myc gene, bringing along transcriptional modifiers, mediator complex, and ultimately driving wild-type MYC expression in T cells. However, in instances where this interaction becomes oncogenic, we therapeutically knock this interaction down and instead see the emergence of a new interaction between enhancer 2, the c-Myc gene, leading to a drug-resistant MYC expression phenotype in leukemia. And so taking a naive approach, it's impossible to just look at the linear sequence of the genome and know how the right enhancer for a gene is going to interact the right gene at the right time in a healthy condition and how that changes in disease. And so it's our perspective in the Hansen lab that we need to apply 3D genomics methods to begin to isolate these interactions and their relationships. And so to give you an overarching agenda for today's talk, I'm going to be talking in the first part about RCMC, or region capture microsy, which is a technique that we've developed in the Hansen lab which allows us to capture structural detail at very high resolution in loci we care about. And then I'll segue from there into connecting RCMC to its potential role in helping translate genetic variants to function. And there's two sub-stories here that are both in the relatively early stages of analysis that I'm going to give you some insight into. One is a project with Melina Klauschnitzer here looking at how we can utilize the V2F framework applied to RCMC data to examine metabolic diseases. And the other is a collaboration project with Vijay Sankran, also in the GROW community, looking at how we can utilize RCMC and a V2M framework to understand blood disorders. And so to dive right into some background on RCMC, I know I motivated the need for us to connect variants to functions by beginning to understand how enhancers connect to their cognate genes. And so one of the really traditional workhorses in this field have been chromosome conformation capture technologies, or 3C techniques. The gold standard of which has been this technique called Hi-C, which I'm showing the method for here. And so in a nutshell, Hi-C allows you to cross-link DNA, digest it with restriction enzymes, ligate the free-floating ends, and then create a library that allows you to reconstruct structural information. And Hi-C has been excellent for finding higher-scale genomic structures. However, when it comes time to resolving fine-scale genomic elements and the interactions in between them, Hi-C has run into some limitations. First, Hi-C is limited in its technique resolution because it relies on restriction digestion. Restriction enzyme sites are not ubiquitously and evenly spread throughout the genome, nor are they all equally accessible after you've cross-linked DNA, which leads to large and heterogeneous fragment sizes. And so if we swap out restriction digestion to digestion with microcollical nuclease or MNase, you're able to turn this disadvantage into an improvement in technique resolution, and you come out with micro-C. On the other end of things, having a genome-wide pairwise method creates a massive contact matrix that you need to populate with sequencing data. Even with all of the advances that we've had over the last 2 decades in sequencing depths, it's still prohibitively expensive to get a high resolution level of detail across the genome, several tens of thousands of dollars in sequencing. And so if you fail to sequence adequately deeply enough, you run the risk of having a sparse matrix, which ultimately makes it difficult to extract structural insight. And so one way we can take on this challenge is to trade away genome-wide information and instead concentrate our sequencing reads to loci that we actually care about. And so this effectively reduces the size of the contact matrix, densifies your data density, and is achieved through capture. And so if we stitch these two together, micro-C and capture, we come out with region capture micro-C, which is a technique that we developed earlier in my PhD work in the lab. And so I'll just very quickly walk you through what RCMC is. In design, it's quite similar to Hi-C, and so you take chromatin or nuclei, you cross-link them with 2 different cross-linkers. You'll digest them with MNase, which gets you roughly nucleosome-sized fragments homogeneously throughout the genome. From there, you can label the free-floating ends, ligate them to their spatially proximal neighbors, and then do a couple of purification steps to ultimately come out with a library. So this library is an extremely high complexity micro-C library that you could sequence right here and then, or you could perform target enrichment for using probes for regions you care about, and ultimately, after one final round of amplification, put that on a sequencer and come out with contact map data. And so before I dive into the kind of insights we can get from RCMC data, I do want to recognize the fact that not everyone has a background in 3D genomics, and so I'm going to take 60 seconds and give you a crash course in how to read this data. I'm still learning. And so 3D genomic structures are visible on contact maps, which are really just matrices mapping how frequently 2 points on the genome interact. And the darker a point is, the higher the interaction frequency. And so here I'm showing you a representation of a contact map. The diagonal represents the linear genome. The areas above and below the diagonal are the contact matrix, and I have some DNA annotations shown below. And so if I want to try and understand what this enriched dot means biologically, I'll move horizontally and vertically until I hit the linear genome. From there, I can drop down to the DNA annotations, telling me that this is the interaction frequency between RBM19 and MED13L. And then I can stitch that information together with 1D genomics data sets like protein binding from CHIP data sets or epigenetic marks and RNA-seq data. What's nice about this kind of visualization as well is that you can have 2 different data sets viewed at the same time, one above the diagonal, one below. So in this case, we can have data set 1 with the contact present and data set 2 with the contact absent. So now that you all know how to read contact map data, let me show you what our prior work using RCMC really allowed us to do. And so here, I'm showing you above the diagonal RCMC data that we generated in MESCs. Below the diagonal, I'm showing you the highest resolution available Hi-C data set in MESCs, generating from over 3 billion unique contacts. And here at 250 base pair resolution, you can see just the sheer amount of greater structural detail we're able to resolve using the RCMC data, which also then allows us to resolve interactions that weren't previously visible. And these interactions allow us to link different genes within the locus and really see these functional relationships. On a quantitative level, if we just simply quantify the average number of unique contacts in each contact bin as a function of distance, you can see that RCMC is 1 to 2 orders of magnitude richer in contact density, which just goes to underscore that we're able to generate these very data-dense data sets at loci that we care about and begin to resolve relationships between functional genomic elements that weren't previously visible. And so I've given you a little bit of background on RCMC, but this is a V2F symposium. We care strongly about how we can create a linkage between genetic variants and structural variants. And so I'm going to dive into the first part of my 2 stories on this front. And so looking first at a story around a project where we're looking at the genetic basis of obesity. And so you may be familiar with the FTO locus, which is a heavily studied and well-characterized locus that is strongly associated with obesity risk and is known to have long-range enhancer control. And so if I show you here the locus and an LD plot in specifically showing you some variants of interest within the FTO intron, you may naively assume that if there is some phenotypic consequence associated with these introns, that it would likely impact the genes that are in linear space the most close to the FTO gene, so like RBL2 or AKTIP. But interestingly, prior studies have found that the difference between the non-risk genotype and the risk genotype in terms of mRNA expression primarily manifests at 2 different genes further away from the FTO intron where this variant exists. And so I've highlighted those here, IRX3 500 kBOA and IRX5 1.2 megabases away from the intron. And so we know that this intron has a strong association with obesity relating to a 70% increase in obesity risk. We also know that it has some sort of expression correlation in terms of having the variant and having some impact on IRX3 and IRX5 expression. So the question began, how can we begin to reconcile this in terms of, is there perhaps a structural role in terms of contact being played to link this variant to these 2 different genes? And so I've been extremely lucky to work with a set of fantastic collaborators, Melina, Joaquin, and Hassam. And so I'm going to be diving into some of the work that we've done together. And so to just motivate the system in which we do these studies, HWAT cells, or human white adipose tissue cells, are taken from human neck fat, and you can immortalize them and then grow them over time and collect different time points of differentiation through adipogenesis. And so here we collected time points for day 0, day 3, and day 14. And what's striking is that as these cells go through this differentiation protocol, they exhibit striking phenotypic change, not only morphologically in terms of how they present and how are visibly discernible as they progress through the differentiation, but also in terms of their genetic activity, which is a promising system for us to apply RCMC in. And so we applied RCMC specifically at 18 different loci, one of which was the FTO locus. And so here I'm showing you this data. In the left third of the map, we have day 0 data, and I've just annotated below the many different contacts that we see, and in particular, the IRX3 and IRX5 loops with the FTO intron. And then we have day 3 data in the middle and day 14 data on the right. I've also noted right below the contact maps the location of the variant of interest. And then there's a series of 1D genomics data sets and some genomic annotations at the bottom. And so taking a quick qualitative look at this data, clearly we want to zoom in on the central domain where the FTO, IRX3, and IRX5 interactions are happening. So we zoom in and just take a look across the transition. Maybe a little hard to tell on the screen, but there is a significant weakening of the domain in terms of the insulation of the domain and the average degree of interaction within the domain as we go from day 0 to day 3. And as a result, the interaction intensity between IRX3 and IRX5 and the FTO intron also happens to diminish. And so what we can do is we can take this qualitative analysis, move it into a little bit more a quantitative space. And so I'm just showing you on the left a representation of the contact map that you've already seen. And then a pretty low-level analysis of the contact strength as we move across this transition, normalized for the expression in day 0. And so it's clear that both FTO IRX3 and FTO IRX5, those contacts diminish sharply in strength going from day 0 to day 3, and they recover slightly going to day 14. Interestingly, if we look at gene expression across that same series of time points, we see that the IRX3 gene monotonically decreases in expression across the transition, whereas IRX5 actually increases slightly for decreasing. And so at a first glance, it doesn't seem like there's a strong corollary between contact strength and gene expression, but we can turn to a well-established model in the field, the activity by contact, or ABC model, which allows us to integrate both enhancer activity and contact intensity. And so briefly, if you're not familiar with this model, it posits that if you take the product of the activity of an enhancer, which we can measure through H3K27 acetylation signature, and the contact intensity using Hi-C or RCMC as I've been describing, then you're able to create a score which allows you to relate the impact of that enhancer's likely activity upon a cognate gene. And so in this case, you're seeing two different enhancers that vary differently in their activity and their contact levels. And it allows you to begin to approximate how changes in activity or changes in contact intensity may relate to changes in gene expression. And so using this approach, we first figured out what the H3K27 acetylation signature looked like across the transition at these sites. And as you can see, it monotonically decreases for both IRX3 and IRX5. And then applying this first order ABC approximation for the interaction between FTO's intronic variant and IRX3 and IRX5, we find that IRX3, which is closer, 500 kb away, seems to be more reliably fitting the model and what we would expect as changes in normalized contact intensity multiplied by the H3K27 acetylation signature change in relation to RNA expression. But interestingly, the IRX5 interaction doesn't seem to be as reliably predictive of IRX5 gene expression, which from an early stage analysis could be due to a variety of other reasons. One, IRX5 being 1.2 megabases away. Two, the domain upon the edge of which IRX5 sits is the domain that weakens, which may collectively shift the enhancer burden of regulation over the course of the transition. And so this is something that we're looking more deeply into. But for an initial proof of concept, I think we were satisfied that we were able to generate RCMC data using these systems and begin to ask interesting questions that related structural intensity to gene expression. And so moving into some new directions, some current analyses that we're working on. One, we're looking at some of our other loci, as I mentioned, because RCMC is able to deeply reduce our sequencing burden. We're able to capture many loci across many conditions in parallel, and so we have 18 total loci. And so this is another one of those loci. Interestingly, here, there is a very strong morphological change in terms of the emergence of a series of contacts within this domain as we move through the transition. And so structurally, that's all fine and dandy, but what does this mean from a V2F framework? And so there's two key variants which I've highlighted here with the red arrows. And you'll notice one of those variants sits at the promoter for the PPARG gene, and the other variant sits just downstream of it. And so if you look carefully at where the promoter for PPARG sits, where that variant sits, you'll notice that there's actually an emergence of a domain that connects the front sort of third of the PPARG gene that emerges across this transition, indicating that there is some amount of perhaps internal regulation of PPARG that's occurring. You also see some contacts emerge upstream where PPARG is interacting with genes that are in the local genomic neighborhood, like TIMP4 and SYN2. And then interestingly, you also see the creation of a new domain that has been subdivided out of the original day 0 domain by the time we get to D14. And so beginning to quantify the emergence of several nested contacts and changes in overall domain strength to individual genes, individual variants, and their relative interactions is something that we're actively looking at because we do have this rich data set. I'll also note, hearkening back to the fact that FTO is known to have a risk and non-risk genotype with a significant implication on the phenotypic end for obesity risk, If we compare the risk allele, the CC allele, against a non-risk phenotype, there's a clear difference in relative mRNA level. And so what's nice is because RCMC is a sequencing-intensive method, we're able to dive into the data and separate out the data that comes from a donor that has one allele versus a donor that has another. So we're working to dive back in to create separate contact maps that are haplotype separated to then allow us to create a more stringent interaction between not only variant and function, but specifically haplotype and function. And so just to summarize this part of the talk, we're working on these haplotyping calculations, looking at these 17 other loci. Many of these other loci are implicated as type 2 diabetes risk loci, which allows us to step into a whole other space for metabolic disease. And then our ultimate ideal goal in this work is to develop a scoring framework that would allow us to relate structural data that we find to be able to do some amount of prediction for what kind of effect a variant would have. And so to move into the last fraction of my talk, I'm going to be diving into how we've been using RCMC to understand the genetic basis of blood disease. And so you may be familiar that fetal hemoglobin is a key modifier of sickle cell disease and thalassemia. And previous work from Vijay Sankran's lab has determined that there are several key loci of interest that seem to be determinants of fetal hemoglobin levels. And so the question is, like, can we use RCMC to identify the key regulatory mechanisms and interactions at play in blood disorders? And so we have a system, also a 3-stage differentiation system, where we take progenitor cells and differentiate them to proerythroblasts. And we've captured 6 different loci, 2 of which I'll show you data for. And again, I've been extremely grateful to be able to work with some fantastic collaborators on this project. And so I think it is important, before I dive into the data, to clinically motivate why we care about this, because this is the V2F Symposium. You may be familiar with Casgevy, which just a few months ago became the world's first approved CRISPR gene therapy. And it's intended to treat sickle cell disease and thalassemia, and specifically it targets a GATA1 binding site within the BCL11A gene. But what's surprising is that despite it having achieved clinical approval, it's not very well known how Casgevy modifies the chromatin landscape around the gene, how it impacts the interactions of the genes, their expression. And so this is an unmet need, which if we're able to understand mechanistically how Casgevy acts successfully as a therapeutic, we may be able to better replicate that success for other variants of interest. And so here I'm just showing you some RCMC data showing the BCL11A locus with some GWAS hits below. And you can see that those GWAS hits strongly cluster in the BCL11A gene. So if I just highlight that region, it's perhaps unsurprising to you that where Casgevy actually targets, its motif is within this region. And what's striking on a structural side of things is that we see a strong emergence of a contact that is intra-BCL11A throughout this transition. And so if we zoom in a little further, you can see that with just a little bit more clarity. And so if we run the same analyses we ran before, looking at contact strength, we see contacts, once again, increase in strength before decreasing from P2 to P3, and the gene expression monotonically increases. And so when we add in the H3K27 acetylation signature and we generate our first order ABC model, once again, we do see a positive correlation between the product of the contact intensity and H3K27 acetylation signature against RNA expression. But it does seem to be that there is a slight misalignment, and so possible explanations for this could be other enhancers playing a greater role as we move through the transition, or potentially a temporal delay between when contact intensity and H3K27 acetylation signature change and when that propagates to changes in RNA expression. And so just to wrap out with some new directions, looking at that same locus, you can see the emergence of some contacts with a downstream gene called PAPOLG. And using that first order ABC model here, you can see, again, it seems to be already a reliable pretty first stroke representation of how we may be able to predict RNA expression, in this case, potentially saturating as contact strength and H3K27 acetylation signature grow. And just looking at this other last locus, As we zoom in at the variants of interest right here, it's interesting to see how this locus actually sees a large loss of interaction as opposed to the other loci that saw large gains of interaction over the course of the transition. And so interested in quantifying how this changes as well. And so we're currently looking at how we can intersect the structural hits with BCL11A CRISPR screen hits, doing some statistical fine mapping to really hone in on the variants that are most causal for phenotype, and then really closing the door on the Casgevy story to really define the mechanism of action there. And so I hope I've shown you today that RCMC is a tool that allows you to generate high-resolution structural data, and that also it's a promising tool in the V2F toolkit to help you understand variants and how they may relate to function. And so I just would love to acknowledge the Hansen Lab, Anders, my PI, and our wonderful collaborators at the Broad, and to all of you for sitting through my talk.
It's a very interesting talk. So we have some time for questions. We'll start with Jason.
That was awesome. I have a really basic question. For me, genome structure and accessibility or continuous of subtle is like a chicken and egg kind of thing. I'm just curious if you've done perturbational experiments where, say, you add RNA polymerase inhibitor and see if there's what happens to genome structure in an attempt to get at if it's causal or if it's correlated to gene expression.
Yeah, excellent question. So I didn't have time to talk about that here, but in our prior work, which you can read in the Nature Genetics paper that I referenced, we did a pair of 2 transcriptional inhibition experiments. So we treated with triptolide for 45 minutes and for 4 hours, and in both those instances, we saw negligible effects on the manifestation of these structures. And that could be due to a couple of different reasons. Potentially, that could be a temporal reason where structure may precede the gene expression. On the other hand, it could also be that mechanistically, when we're at this very fine scale, it seems to us increasingly so that non-loop extrusion, non-AB compartmentalization are really driving interaction between these active genomic elements. And so if there's a different mechanism at play that may be orthogonal to the need for polymerase activity, we believe that could have some explanation for it. And then we have an ongoing project looking at the mitosis to G1 transition to see temporal emergence of contact versus expression. So as we dive into those analyses, I think we're hoping to get that chicken or egg story figured out. Thank you.
Thanks, Viraat. That was a beautiful talk and very exciting directions. I guess on your efforts trying to map, to link the contact maps to the expression output, I was wondering your thoughts about incorporating different modalities of RNA, nascent RNA, like ProSeq, or also now with single cell resolution-based data where you can actually have cells organized in pseudotime and try to match some of those states to actually your contact map and see what could predict it.
Yeah, that's an excellent question. I think this has been sort of a largely underexplored frontier on our end. I'm a structure-first guy. I'm coming from the 3D genome end of things, and I think that as I learn more and more about how different 1D genomics datasets may be able to be integrated into the model we hope to build, the scoring framework we hope to build, I think it is important to recognize that there is this wealth of data that already exists. Granted, for some of the systems that we're studying, there may or may not be some of these datasets that already exist, but I think because RCMC acts in a way as kind of a hypothesis-generating sort of discovery approach where you can be like, oh, here are some interactions I hadn't considered maybe of causal interest, right? I think as you go through that discovery phase of characterizing what you're seeing, I think having insights from 1D genomics datasets like the ones you've mentioned is very helpful, and I think I just need to learn more about how I can best integrate them into my own analyses.
So we'll start with the online question and then with Brad.
Yeah, we have some online questions. I try to summarize them in 1 or 2 questions. So 2 questions were about scalability. Mm-hmm. How would you scale up RCMC for machine learning applications? And then how many loci do you think you could realistically be profiled at once in RCMC design? And the other one is how does it compare cost-wise to high-C
Yeah.
And does it work on single cell level?
Okay, excellent question. I will bundle my answers for those. So back of the envelope calculations, say you want to capture a total of like 7 to 15 megabases of the genome. That could be one locus, that could be 7 to 15, 1 megabase size region, it doesn't matter. You can get probes for that for, let's say, like $4,000 to $6,000, and those probes will be good for 12 reactions. Each reaction you can multiplex 8 microC libraries into. So about 96 microC libraries worth of target enrichment you can get for $4,000 to $6,000 for 7 to 15 megabases of the genome. So assuming you use all of them, that's like $50 per library, per experiment for target enrichment. And then the sequencing dividends you get are insane because the RCMC versus Hi-C dataset I showed you, the Hi-C dataset had 3 billion unique context genome wide. The RCMC dataset had a third of that, but then was still 100-fold deeper in terms of data density. And so if you're going to be profiling some loci across many conditions or many perturbations, I think it's very worthwhile. If you're only going to perform target enrichment once and not use those probes multiple times, then I think the cost-benefit analysis trades off. So I think you save money on sequencing costs if you're going to be using the probes a lot. I definitely forgot one of the 3 questions. I think the first one.
Scalability.
Scalability, yes. So in my prior work, we were able to simultaneously do 5 perturbations, so 5 out of the 8 possible in tandem. So I think what's nice about a technique like this is because you're saving money on sequencing costs, you can begin to ask those interrogative biological perturbation questions that I think are really important for the field to begin to move from a largely observational standpoint, which that's my bone to pick with 3D genomics. That's been largely observational for 2 decades, and to really move into a functional space. And so I do think it's scalable. Is it what I think the Broad would call high throughput? No. Is it much higher throughput than I think people in the 3D genomics field have traditionally been used to? Yes. And so I think it's the perspective you take.
Well, one last question from Brad.
Hey, it's great to see the story developing. I have a question about the IRX locus.
Yeah.
If you look at IRX5, the promoter, there's a stripe coming from the promoter that extends for very long range contacts and a stripe, which might reflect, I guess, extrusion of one strand while the other's fixed, and maybe explain the long-range interaction. But also, I think Alistair Bodinger's has an alternate hypothesis that maybe this is stacking of CTCF sites. So I'm wondering what you guys are making of that structure.
That's an excellent question. Yeah, and so there are a couple ongoing hypotheses that we've bandied about in the lab for sort of flames or stripes like that. On one hand, you can have basically one end of the extrusion anchored by, say, if there is a CTCF site there or a complex, maybe a polymerase. I know that there's ongoing work in the lab working with Leonid Murney's group in the physics department, trying to understand at like a larger scale how we can characterize like what is binding at the sites that are known to have flames. I do think that it poses an important challenge for us to be able to apply a framework because how do I quantify the strength of a smear versus a strength of a dot? I think the strength of a dot is more logically intuitive in terms of traditional calculations of strength in the field. But for a smear, there's not really a widely accepted sort of framework for like how you necessarily quantify that in its totality. And so I think it's an important question, one that we do need to solve. And I think the current hypotheses lie along the lines that you had mentioned.
Okay, I think we're gonna finish this session. We're gonna have 1 hour of the lunch break.
Yeah, we are 30 minutes behind schedule, but we keep the 1 hour lunch break. So please enjoy the lunch, and thank you so much again, Viraat.